since it's a really straightforward topic, I thought Kert from Berry Consultants could cover it in 45 minutes. And please direct your attention up to Kert.
All right. Thank you for having me. I am actually going to try to be as simple as possible. So I'm going to start with some pretty basic examples. I'm going to do trial simulations by talking about flipping coins and talk about how that has some semblance to reality. My talk was originally way too long, but then Bill Garson gave about half of it yesterday. So I was able to get rid of it. We actually had the same slides through complete not trying to get together and do that. So quick idea, this question actually came up yesterday. Suppose you have 200 subjects. You want to do a dose selection trial, you got to pick among 4 doses. Trial 1, I can allocate 50 patients to each of the doses. Nice, standard, fixed trial. Run the trial out, pick whichever one is the best. Potentially I can move on to phase 3 depending on how good it is. Somebody comes in, asks a reasonable question. Can I have a control group? Sounds like you would ordinarily have a control group. In a lot of cases, especially in phase 2, you might not want to. If the comparator that you're going to be eventually comparing to in phase 3, if it's sufficiently well known and there's a literature where it has consistent properties, if you're running a small phase 2, you have 2 competing goals. The control group will help you make the go-no-go decision, but you also have a dose selection that you have to make. The subjects that you allocate to the control are not helping you make the dose selection. So there's issues in there, and if you watch pharma companies, a lot of times they won't enroll in a comparator into phase 2 simply because they already feel they have a good idea about how it would behave. And you can show if the phase 2 is small enough that actually makes sense, however counterintuitive it may be. So trial 2, we could run 40 subjects per dose, 40 on control, and we get more information on the control potentially, but we have less subjects on the active doses. So that's an alternative design. So the question is, can we be clever? And can we figure out a way to get the benefits of having 50 subjects on a dose, but still get our control arm in? And so you can do things in the middle of the trial. And again, this goes back to what Michelle was saying. As long as you pre-specify what you're doing, you can understand the properties of it. So it's important to say in advance what you're doing. But I can do the following. I can enroll 25 subjects on control, each of the 4 active doses. That's 125 of my 200 subjects. So at that point, I can conduct an interim analysis. So I look at the data. It doesn't mean necessarily I personally, actually ironically, it would be me personally. You, as the PI, you want to avoid operational bias. You don't want to see the data. You don't want to have any idea that might change your behavior. I, as a statistician, might go and look at the data and can make a decision. This was pre-specified in advance, so I'm not making any clinical judgments. I'm following an algorithm. It's set at 125 patients. Open up the data, have a look, and do the following. And so I could get rid of the 2 worst performing doses. So now they're gone. The notion is we're 125 patients in. At the beginning of the study, we didn't know which of these 4 doses was best. If we're basically halfway through the study, we may be able to start telling you which doses are worst. No point in allocating anymore. This gets back to what Roger was saying about trying to minimize risk to patients. If you have 2 doses that they're really doing worse, would you want to be allocated in a clinical trial? Hey, we kind of know these doses are bad, but we want to know which one's really bad and which one's just kind of bad. So the usual question you want is which one's the best. So let's get rid of the worst ones. We can enroll 25 subjects on the control arm. The 2 doses that are left, I've used up 200 subjects. And now what I've done is I've gotten 50 on the control, and I've also gotten 50 on the 2 selected doses. Hopefully those are the best. So at some level, I've manufactured an extra arm, and I haven't increased the size of the trial. Now there are costs and benefits to that. You could reasonably worry about things like how often do I drop the best dose early? At that interim analysis, what if one of the good doses actually ended up performing badly for 25 subjects? how often do I mistakenly drop it? Very reasonable question to ask. How often does the fact that I have a control arm actually help me make better decisions? So how often do I make a better go, no-go decision? Reasonable thing to ask. I've got 3 designs. I can do 50 on each dose, nobody on control, 40 on each dose, including control, and I've got this adaptive design that gets rid of some in the middle. I could ask that of all these designs, which one is the best for what I want to do. How often do I select the best dose at the end of the trial? So if I have 50 per dose, I'm presumably going to do better than 40 per dose. But the question is, how much better? What did I gain or lose by this? So you can do a lot of very clever things in a design. If you think information is going to be accruing and you could act on that intelligently, you can build it in. Most of the time, this is not considered in a lot of trials. So why not? Why do we always consider fixed trials? Big reason is we've always considered fixed trials. At some level, that's fundamentally the reason is this is what's done. It's easiest to calculate type 1 errors. All the properties that involve, do I have a regulatory nice trial where I can say this trial is type 1 error controlled at 0.025 or 0.05, I can do the math with a fixed trial. Logistically, these are simpler to implement. If I sit here and say I'm going to do 50 patients per dose, I run it to the end, I know how to handle that. This interim analysis, how do I do it? So I have to get another statistician to perform the analysis. I have to tell sites to stop giving certain doses of the drug. How do I make that happen? How do I make sure all my sites get me the data in time to perform the interim analysis? So if I get to 125 subjects and all the sites go, hey, it's going to take us another 3 months to really clean the data, then this is not so practical. By the time you get it, you've enrolled your 200th patient. You don't want to pause a trial, so you got to make sure you get the data quickly. So logistically, this would be harder. Unfortunately, we all know that phase 3 trials are failing right and left. So part of the FDA's critical path initiative acknowledges that while adaptive designs might be harder, they're also in many cases better. Not every case, and perhaps not even a majority of cases. We'll talk about it in places where they're bad. But in a lot of cases, this is a mechanism for speeding drug development. So lots of verbiage on the FDA website about more efficient trial designs, about innovative clinical trial design that make it possible to develop accepted protocols for smaller and smarter trials. The smaller, what I just said is I was able to put an arm in there. Smarter in the sense that we use information that's accruing through the trial to make the trial better. If there's something you didn't know at the beginning of the trial, but you think you might have a good idea of halfway through, you can make use of that information. Change your trial. Now, you can't look at the data, but you can imagine a thought experiment where what kind of decisions might I make? So, obvious decisions you might expect a DSMB to make. If one of the doses is unsafe, get rid of it. Well, what if one of the doses is just terrible and doesn't work very well? Good reason to get rid of that as well. You can consider designs. Some stroke studies are interested in the size of the stroke. Suppose we find out a drug does not work for particularly large strokes. Stop enrolling those subjects. If there's no evidence that it helps, we can go ahead and focus our enrollment on people we actually think we are helping. So at some level, we talked about adaptive trials. You should keep in mind that all drug development is adaptive. But kind of the analogy to make is if I want to get from one part of a downtown to another, what I could do is I could say I want you to drive 3 blocks, then I want you to turn left, drive 2 blocks, turn right, drive another 2 blocks. You've got a destination. So you can think of that as a phase 1 trial, a phase 2 trial, and a phase 3 trial. What we tend to do is hop in a car, close our eyes, hit the gas, hope that when we get 2 blocks down we haven't wrecked into anything, hope that the world still exists, all the streets are still there, and then we go, okay, let's get reoriented, get everybody turned the right direction, pick everything we need to do, and then we get back in the car, we close our eyes, we blacken out the windows, we drive again, and we hope that all these steps have actually worked. So you can think of adaptive trials, one of the goals is to drive with your eyes actually open. So if it looks like your car is running off the road a little bit, so you need to pre-specify, you need to say how you're going to do this. But if things start coming at you, if a cow falls in the middle of the road, drive around the cow. So if you get anything else out of this, drive around the cow. So Bill Barton talked about this a lot yesterday. So the kind of things that you want to worry about when you design a trial, what might go wrong at the end? So you can imagine that the example that I was aiming at, certain doses were bad. So I wish I had given more patients the following 2 doses. I got 4 doses. These 2 were terrible. Which of these 2 is best? Boy, it would have been a lot better if I hadn't, in some sense, wasted all these subjects on these other doses, because the real question now is which of these 2 is better. If I could have learned that as early as possible, I don't know if I necessarily would have settled this, but I'd have a better chance if I had allocated more patients to those promising doses rather than every dose. I talked about the size of the stroke. Maybe I found out my treatment didn't work for certain people, so the inclusion and exclusion criteria. Interesting example, got a p-value of 0.06. What if I had just enrolled a few subjects? So we had the following example. This actually happened. Company runs a trial. They get a p-value of 0.06. So this is bad because 0.05 is absolutely magical. So 0.049, you found the fundamental truth. 0.051, your drug is no effect. So anyway, obviously it's not quite true. But 0.06 is bad. You will not get approval on 0.06 unless you really can convince people that something odd happened. So what did they do? They went, oh, we just underpowered our study. Let's go run another trial. So they went and ran another trial. Can you guess what their p-value was in the other trial? 0.07. So it was close. So they're still close. Now bear in mind, their point estimate's going in the right direction. They have a trend. So you could argue they ran 2 underpowered trials. So then, can anybody guess what they did next? They went to the FDA and they said, hey, can we pool the data between the 2 trials? Because then our p-value is 0.02 or 0.03 or whatever it happens to be. Can you imagine what the FDA said? No. Good. Are you the FDA? No. So anyway, the FDA said no. With reason. So this wasn't pre-specified in advance. It's unclear, what all the multiplicities are. This gets back to what Michelle was saying. And at the end of the day, you have not run a trial that has had a significant result. You're trying to manufacture one. So one thing to consider, though, is if you had thought in advance. So what would I do if my trial has a p-value of 0.06? If the answer to that question is I would run another trial, you might as well build that into your current study and actually say, if I get to the end of this study and I have a close but non-significant p-value, I'm going to extend this study for another 100 subjects or 300 subjects or whatever is necessary. Now, as Catherine was mentioning and as Michelle was mentioning, you have to account for the multiple looks. So it's not a panacea. But what it will do is you can get a power benefit out of that. So you can increase the power of your current study, and you built in the fact that I was going to spend these subjects anyway. Running the first trial and then going, hey, let's find another completely separate one is not the best strategy. Building that in advance, pre-specifying it, controlling type 1 error for the process from the beginning will let you have a better chance of success. You can imagine you got a p-value of 0.000001. Boy, I should have found that out earlier. So as this was noted, this doesn't happen very often. There really aren't that many trials that are this great. But it's possible, so maybe you do want to have the possibility to stop early. The last one happens a bunch. Suppose you power a trial on a bad assumption of variance. You say, I need 100 subjects because I think I have a standard deviation of 5. Well, you run the trial, you find out the standard deviation is closer to 7, and in fact you needed 200 subjects instead of 100. So there are ways to adjust for that. If you have uncertainty about the standard deviation, you can run your trial partway through and do what's called a sample size re-estimation, which basically says, based on the information I now have, what is the standard deviation? How big should my trial be? The answer at the beginning of the study was, I don't know. The answer later on in the study, I have a better idea of the standard deviation. I now know better what the trial ought to be. Let's try to get it closer to that. Again, you've looked at the data. You have to do an adjustment. You need a good statistician who's able to tell you how to do this. But it's a way of making your trial robust to the uncertainty you have. You don't know what the standard deviation is. You need to figure it out. All right, so I talked a little bit about this. Roger hit this as well. Equipoise. So a question. Would you rather not be the last patient enrolled in a clinical trial or the first person afterwards? If you're the last person into a clinical trial, they flip a coin to decide what treatment you get. Virtually, but essentially yes. The main thing I will ask about a clinical trial is if your doctor is choosing your treatment on the basis of a flip of a coin, wouldn't you want the coin to get smarter with age? So later in the study, we're going to talk about something called, we'll talk about something called adaptive randomization. And the goal is essentially later in the study, you make use of the information you have, and you say, because these doses, these treatments are doing better, you're going to be more likely to be randomized to it. Roger, it's you and Don have the JAM editorial on this, right?
And Will.
And Will, okay.
Actually, Will's the first author.
Okay, so Will is the world's expert on this also. But this is something that's actually good for consenting patients. It basically says, after we learn that a treatment is, we're not going to give it to you anymore. This is a good thing. All right, so adaptive trials are by necessity complicated. They involve interim analyses. They involve changes potentially in the sample size, changes in the dose schedule, who's involved. Ordinarily, it's nice if you go to your local statistician and you say, I want to run a fixed trial, they can call up a computer program and they can go, hey, I click this button, I click this button, I click this button, I have a sample size. I have a power, I have a type 1 error, I can tell you what are the odds certain things happen. If the trial gets complicated, you can't do this anymore. So you end up in the difficulty, there are no computer programs, there are no formulas anymore. So if you ask, I talked about this adaptive trial that dropped 2 of the doses. What's the probability that I pick the right dose? I can't sit on a piece of paper over lunch and tell you what that is. So how do I find it out? So let's think of a very simple question. Suppose I flip a fair coin twice. What's the probability of 2 heads? So I'm now the proverbial horrible statistics teacher because coins have nothing to do with reality. So, anyway, so I did this once and I had a student, so I will say one of the students in this particular lecture gave me a marijuana brownie. Yeah, I kid you not. And they were pretty loopy for the whole class. But the other one, it was a coin experiment, and it actually gave me a 2-headed coin. So I actually have been caught by the heads. Thing. So anyway, if I flip a fair coin, I ask, what's the probability of 2 heads? It's easy. There's a formula. 0.5 times 0.5. You get 25%. Easy. Okay. There is another way to do this. You can sit very boringly and go, I'm going to flip 2 coins. I got 2 heads. Great. I'm going to write that down. I'm going to do it again. Flip 2 coins. I got a heads and a tails. Do it again. Tails, tails, heads, heads, over and over and over again. If I do this enough, eventually, and I just count how many times did I get 2 heads, I'm going to get to 25%. Now, I am never going to do this. I can have a computer flip coins to kingdom come. So, I mean, you can easily have a computer replicate this thing 1 billion times in not much effort. Now, we like the top formula. It's a formula. Some mathematician came up with it. It feels intellectually savvy. The bottom thing feels, ugh. Okay. But it's one thing that's really nice about the bottom. In hard problems, you can still do the bottom. And it still works. So it's essentially robust to the complexity of the problem. The trial that I was going to do before, where I enrolled patients, had an interim, dropped 2 doses, I can do that on the computer. Generate random numbers, generate 125 subjects, tally up which 2 doses I would have dropped, enroll the next 75 patients, see how often I pick the right dose. I can do that over and over again, simulate one trial, another trial, another trial, another trial, and so on. And I can take that long list of trials and just count up how many of them won, how many of them picked the right dose, how many of them eliminated the best dose at the interim analysis. Don't want to do that. So I can tally all these particular things, and I can compare my 3 designs. So if trial 1, which was fixed, trial 2, fixed, had it in control, trial 3 that's adaptive, what I can do is I take all 3 of those, if I can do any of them analytically, if I can get formulas, I will, but any of those trials I can simulate, and I can ask which of those 3 designs is most likely to win, which of those designs is most likely to pick the right dose, which of those designs is most likely to make the right decision going to phase 3. And so the reason trial simulations become so important is as we consider complex designs, this is the only route available to us to do those kind of calculations. So we're making use of the fact that if you sample something enough, all those proportions and means converge to what the truth is. So it's a very brute force method, and one of the reasons that you see more of it now, of course, this is true with Bayesian methods in statistics. Over the last 30 years, computing power is vastly bigger than what it was. All these things are now feasible and becoming easier and easier by the day. So I think this is what I said. So let me give an example of another kind of adaptive design. This is called adaptive randomization. It's a common dose-finding design. You begin by enrolling a certain number of subjects on each dose. This is usually called a burn-in. So, for example, if I had 200 patients, I might enroll 25 patients per dose. This would be the burn-in. Haven't enrolled everybody, but some set fixed number. Then what I do is I start looking at the data periodically. And what I did in my first example is often called an arm dropping example. You look at it and you say, these 2 doses are bad, get rid of them forever. In adaptive randomization, you look at the doses and you say, which ones are the most promising, which ones are the least promising. And what you do is you change your allocation probabilities. So beforehand, we just got rid of the doses completely. But what might happen here is if 2 of the doses look particularly promising, I may allocate 40% to each of those, and maybe only allocate 15% to a third and 5% to a 4th dose. And I'm allocating the most patients to the doses that look best right now. Now, if you're asking a reasonable question, what if you get rid of a good dose too early? That's what the trial simulation's for. Did that happen? You can simulate the design, make sure you avoid that risk. Let's see. I should also add, you can do this on the basis of efficacy and safety. So if you have some kind of trade-off between efficacy and safety, you can incorporate that. So unsafe doses don't get patients because they're unsafe. Non-efficacious doses don't get patients because they don't work, and you try to find that sweet spot in the middle. So here's an example of how this works. Simple dose-finding trial, 3 doses. I have a control in this one. Dichotomous outcome, so everybody's just going to give a yes or no at the end. My first design, trial 1, I'm going to do a fixed trial. I'm going to allocate 160 patients to the control, and I'm going to allocate 80 to each of the active doses. You may allocate equally. I'm allocating more to the control. I'm going to want to make 3 comparisons. I'm going to want to compare every one of the active doses to the control arm. The control's involved in every one of those comparisons, whereas each dose is only involved in 1. So really pinning down the control arm has some more value because it's used, that each of those subjects is used more often. So that's trial 1. Trial 2, I'm going to do this adaptive randomization, not going through all the details, but I'm going to burn in 40 patients on the control, 20, 20, 20, and then I'm going to adaptively randomize, and I'm going to change the ratios every 50 subjects. I think when I edited my slides, I got rid of the fact this is a 400 patient trial. So I'm going to maintain 40% control. So as I enroll, I'm going to gradually be trying to move towards the good doses and get away from the bad ones. Go up here. And I'm going to declare success at the end of the trial if the p-value of the best dose is less than 0.025. So I'm not taking a penalty here yet for the multiplicities that Michelle talked about, because I actually want to talk about what they are. If I do this, hopefully everybody can see the bottom, when you do trial simulation, you want to make sure that you simulate things under multiple scenarios. Suppose no dose works. How well does my trial work? Ideally, it never declares success. You don't want to make a type 1 error. If my drug does work, so that's my null. If my drug does work, that's this all good scenario. So that's 30, 50, 50, 50. So it actually doesn't matter what dose you get. You've already hit the plateau. It certainly happens more often than you think. So that's one situation. I want to make sure I have high power. I want to make sure that this thing says it works. I'm also going to consider this high good scenario. So you can see the rates are 30 on the control, and then 32, 38, and 50. Ideally, what I want to have happen is as the trial goes along, I want it to recognize that the second and third doses, the 32 and the 38, they're better than the control, but they're not as good as the high dose. So I want to start moving patients onto that high dose as early as possible. If I can recognize that this high dose is good and I get a lot of patients on it, what that will allow me to do is better evaluate safety information. I'll have a bigger database to inform a future trial. So some things that are going on here. You'll notice that trial 1, remember this is the fixed trial, it has a type 1 error rate of 7%. So it's not controlled. I actually tried to control this at 2.5 nominally. So every one of these 3 comparisons, I had an error rate of 2.5%. I put 3 of them together. It turns out I have an error rate around 7. That's the multiplicities that Michelle was mentioning. So I've got 3 doses. I could compare any of them to be good. I end up getting a fairly high rate. If I do the adaptive randomization, interestingly enough, it goes down. So we talked about adaptive randomization as a tool in order to try to find the good doses. It actually has kind of automatic multiplicity control. And it kind of makes sense. So what do you think is going to go badly? If I run a fixed trial and I get a false positive, what's going to happen is I'm going to run off, I'm going to do a subsequent trial, and I'm going to find out that the drug doesn't work, most likely. So I got a false positive, this random high, I go do the second trial, it's not randomly as good anymore, so I find out. If I do this adaptive randomization, if one of those doses becomes a random high in the middle of the study, what do I ask of it? If you're a random high and you're better than the other doses, I'm going to pile patients on you. If you look good early, I'm going to increase your enrollment, which means automatically I'm saying you look good, prove yourself right now. Not in the next trial, prove yourself right now. And what happens is you get regression to the mean. You give enough patients onto that dose to make sure it happens. So you see the type 1 error go down.
Kurt?
Yeah?
So the expected number of patients, even in the adaptive trial, converges to equal allocation in all of those groups. But when it's stomping out those sort of false positive highs, in that individual trial, maybe the second dose is the high, and in that other individual trial, maybe the 4th dose is the high.
Yeah, so this is the average across lots of trials. So it may be in any individual trial, dose 3 got a lot in some of the simulations, dose 2 got a lot in others. So that averages out in the long run. But in any given trial, the doses that were doing well got more. Yeah?
How does this work out if your outcome of interest doesn't occur for 3 to 6 months after the randomization?
So that's a great question. Generally, if your outcome is too late, you can't do this, is the quick answer. So we were asked to do a weight loss trial once, and we were thinking about doing something like this. So we asked 2 questions. We said, when's your endpoint? They said 6 months. Okay, that's good. Can you get any idea of an early endpoint? So Jeremy talked about biomarkers, surrogate endpoints. If you know weight loss at 3 months or even 1 month, it's actually a great predictor. You can do a lot with that. So we talked about, okay, well, we're going to be able to make progress, we get some early endpoints. Then we asked, how quickly are you going to enroll? So they had a very large study. Do you know how long it takes to enroll a weight loss trial?
I have no idea.
Apparently it takes 2 weeks. So anyway, I mean, they were basically going to open up the floodgates and boom, they're done. So this is completely off the table. There's nothing you can do with adaptive randomization in that setting. Now, if you have a study that's going to go for 2 years, even if you have a 6-month endpoint, you can still make a lot of use of this. So we've done, say, spinal injury studies that have about that kind of timeline. So you're going to have enough information early. You can't start adapting at 6 months because that's when the first completers come in. But halfway through the study at a year, you can start making some credible decisions. So it really makes a difference how fast is your accrual relative to your endpoint. And if your accrual is too fast, this is not going to help you.
So is your interim analysis triggered by the number of enrollees? Or like, say one dose has 23 subjects and another dose has 26. And like, how do you decide when to stop, when to do the analysis? Do you, if the goal were 25, do you exclude the extra subjects that have already been enrolled, or how does that work?
So in practice, you would employ some kind of blocking so that things move along in step. So blocking would say, I don't want the groups to get out of whack. I may have dropouts or other issues. But what you would do in your protocol is you'd have to specify that rule. A typical rule would be when the 125th person enrolls in the study, this analysis will take place with whatever data is available at that time. Alternative rules, when the 100th patient completes 6 months, you could do an analysis. You could say, I want to do this every 6 months. In a cardiac study or a stroke study, you might say, I want to do the analyses when I see 20 strokes. And I'm going to do another interim when I see 30 strokes and another one at 40. So you could base it on the number of events. So you can specify whatever works for your problem, but you have to write it down in advance, because remember what we're doing in the trial simulation, we're getting a computer to basically run through multiple trials according to those rules. So they have to be the kind of rules you can program in a computer. It can't be, hey, we're going to start the trial and Harrison's going to feel like an interim analysis at some point. So you need to make absolutely sure and say when this is going to happen. Yeah?
Can you just say a little more about how simulations help prevent you from dropping a good option that seems like in the initial phases of a bad option? I just don't understand how.
So the simulations themselves, the simulations themselves don't, it's not doing the simulations that prevent them. What you would do is suppose that you looked at the simulations and you said, I have a design that does an interim at 125. I could say, it turns out the best doses drop 20% of the time. So 1 out of every 5 simulated trials, oops, the best dose went away. That's bad. So then what you might do is go, okay, suppose I didn't do that interim analysis at 125. Suppose I did the first interim analysis at 150. And now I could look and say, okay, I'm going to have a little bit more data. Now the best dose only gets dropped 7% of the time. So that's an improvement. And you have to start weighing, I've got the power of the trial, the go, no-go decision you have to balance. But what you're doing, the simulations aren't improving the design. What the simulations are doing is letting you see how each design works. And then you have to decide which design you like best. So you would typically compare multiple design options. And that would be changing the interims. Maybe I don't want to drop 2. Maybe I want to drop 1 dose when the 100th patient's in, and I want to drop another dose when the 150th in. I've still dropped 2. I've done the same sample size things, and that may work better. I don't know. This is a lot of our back and forth with clients. So let's keep tweaking and keep making this better. But we want to make sure that by the end we've pre-specified everything.
Do you simulate so when the subgroups are, so that you're gathering your initial, so you have, let's say you have 20 in each arm. So your type 1 error rate is higher at the first iteration of your interim analysis than at any other time because the sample is so much smaller, right? So how do you really know that you're not just by chance just wandering to a wrong dose? I mean, is there an estimate that you have to calculate beforehand to mitigate that possibility?
So this gets back, it's related to her question. This is what you would do in the simulations, is you would try to actually quantify how often you go down these bad paths. So if you're looking about how often do I drop a bad dose, or how you basically are asking, do I have enough certainty to drop a dose at 125? So we would tend to just operationalize that as how often do you make a bad decision. And if you make a bad decision too often, choose another design. So move the thresholds, drop in a different way. You may find out, so to kind of take your question to its conclusion, you may find out your sample size is small the whole way through, and I never feel comfortable dropping anything. So, and that may revert you. You may at the end of the day go, I'm not going to have enough information to successfully drop a dose. I'm going to run a fixed trial. Especially affects Chris's question. If I accrue too fast, I have an issue of I may have 25 subjects per dose, but only 7 of them are complete at the interim. That's another version of I don't have enough information, I can't do this, is why this would come about. Yes?
What if your best dose also has the highest rate of AEs? Not SAEs, but just some side effects.
So that's a great question. What we would typically do is, instead of doing this adaptive allocation, you wouldn't necessarily do it only on efficacy. What you would do is you would have some composite of efficacy and safety. So we've actually, I give a whole other talk on this one topic. So, but we would often show clinicians prior to the trial, suppose that the withdrawal rate for the study was this, and the efficacy number was that. Is that a good drug? Is that a great drug? Is that a bad drug? And we would have them rank those, and we would gradually try to get an idea of what range, what trade-off do you have in mind that makes for a great trade-off, what's a good trade-off, essentially get the risk-benefit ratio out. And so instead of making the decision to do the adaptive allocation or the arm dropping on the basis of just efficacy or just safety, you would go back to that and say, look, we spent a lot of time figuring out what the right risk-benefit ratio was. We're going to drop the dose that has the worst risk-benefit. It might have good efficacy and it has too many AEs, or conversely, it might be safe but it doesn't work. But you can incorporate that in advance. It takes a lot of effort. We've also done this not just with efficacy and safety. We've also incorporated marketing endpoints, for example. So for certain drugs, it's important that it's not clinically relevant, but the weight loss profile of the drug makes a difference. And so that's been incorporated into the process of, okay, clearly we don't want to have cardiac events. That would be really, really bad. We want to have efficacy, but if it comes down to the third tiebreaker between weight loss, we're going to incorporate that in our decision on where we're going to allocate patients. So you can make these quite complicated. But if you're going to make safety, if safety is a big part of your dose selection, I highly recommend you put it in advance. Make sure you go to the doses that are safe and get away from the doses that aren't. I'm not going to get through my whole talk, which is great. Because it starts to lose a bit after a while. So anyway, but we've had the experience where we've gone to DSMB meetings. Now, this wasn't me personally. I don't think it was Roger. But anyway, we've gone in and the DSMB has said, you know, this dose is looking really bad. We think you ought to drop it. And been able to say, you don't have to worry about it. The algorithm automatically dropped it 2 weeks ago. So it's already happened. You didn't have to go to any efforts. So the algorithm was set to do this in advance. The other aspect of this adaptive design, so I also increased the power Now, I don't do much in this all good scenario, but if you look down and where the high dose is good, I get a power increase from 87 to 92, which doesn't sound that much. It actually takes a sizable percentage of patients to get the power to go up that much at the high end. You can also think of this in terms of here, I'd miss a good drug 12% of the time. Here, I only miss a good drug 8%. So I've gotten rid of 33% of my type 2 errors by doing this. The reason this has happened, the adaptive design, if you can look about how it allocates, on average, it gets away from this dose 2 and dose 3. And it allocates 157 patients to the high dose. It figures out in this setting, I want to go to the high dose. In these settings, it tries to explore more. So if some of the doses are really bad, they do go away. More precisely, if one of the doses is really good, it gets more. Yeah.
I apologize if you already said this. With these adaptive randomizations, how certain can you say that the doses that are eliminated would not be found to be effective?
So what we would end up doing, so we would have a situation, so I haven't shown it, but for this particular scenario, we would create another table, and it would say, how many times did this dose get picked as the best at the end of the trial? How many times was this dose picked as the best? How many times was that dose picked as the best? In this trial, what you'd like to have happen is you want it to always pick this dose, because it is the best. That's not going to happen, of course, but you hope it's very high. We would also have, so we consider other scenarios. So suppose that instead of the scenario I have there, I've got 30% on the null, I've got a bad dose 1, and then I've got 50% on dose 3, and 55% on the highest dose. So I'm setting up a situation where the second highest is pretty good, and the highest is a bit better. That's a situation where you might expect that the model is going to make a few mistakes. So it's going to pick the 50 instead of the 55. So what we do is we go, okay, in trial 1, how many times do we pick the right dose? In trial 2, how many times do we pick the right dose? Generally speaking, trial 2 is going to pick the right dose more often. So as for how often, it depends on how close the doses are together, and it's very scenario specific. But generally, you can do better. Doesn't mean you're always going to do perfectly. You almost never do perfectly. For what it's worth, dose selection is really, really hard. So if you have a good trial that has 90% power and you've got 4 doses, how often do you think you pick the best dose?
20%.
It's better than that. So now if one of them is way better than the other, then you can do great. If you have a nice little Emax standard dose response where it goes up, generally speaking, if you incorporate safety, allow for the possibility of a U-shape, it's more like 50 to 60. So now it gets up to about 80% if you say, let me take 2 doses forward into phase 3. Then you do much better. So that's one reason why you don't necessarily want to pick just 1 dose out of phase 2 and go to phase 3. A lot of companies are now taking 2 doses into phase 3, simply recognizing there's a good chance we didn't find the right dose. Either there's an emergent safety concern on the high dose that we didn't know about, we want to back up, or conversely, there could be other reasons. But you don't figure you've solved the dose selection in phase 2. You've just reduced it considerably.
And in that scenario, you're saying the phase 3 is still adaptive, or is it just fixed?
The phase 3 could be adaptive, it could be fixed. So you could do fixed. In a lot of trials these days, it's adaptive in the sense that you start, there's an interim, and you drop one of those 2 doses. A lot of times it may be based on safety, for example. Alzheimer's trials, they often have a very long endpoint, so they may often take 2 doses forward because they're worried about safety, but they feel they've answered that safety question pretty early, and they may drop one of the doses out. So it really depends on your profile for your drug. Brandon?
Is there a way to build in sort of, especially in the early design phase where you're doing dose finding, is there a way to build in multiple endpoints if you're not sure whether a chemical endpoint or a clinical endpoint might be more a higher yield?
Yes, this would probably be similar to the safety efficacy. So the ways that you can build those trade-offs in. You do need, if you're going to do an adaptation or the arm dropping, you're going to need to do that on some one-dimensional quantity of those. So you're going to have to weight them in some way. You can say, for example, I want to do the best, I want to adapt in a way that I put patients on the most efficacious dose, and I put patients on what's called the NED. So the lowest dose, I think, is effective and safe. So you can be clever and try to hit two ranges, but typically you adapt on some weighted average of your endpoints.
How many published trials have used these kinds of methods?
It's a good question. So publications, I'm not sure. We do, what would you say, about 100 of these at any given?
So our group commonly cites that we have designed over 100 Bayesian adaptive clinical trials that have been reviewed by the FDA.
Yeah, we've done more than that. We said that like 3 or 4 years ago.
Okay, well,
If you look back and said this number of the 100 trials, if we had used a fixed design, would not have reached statistical significance. But so you actually quantified the delta. So maybe what's your best shining success? And do you potentially view the adaptive design as ever having been misleading on the opposite extreme?
So well, the simulations ought to lead to that to some extent, because you can see in advance how often the fixed trial would have won and how often the simulated trial, or how often the adaptive trial would have won. It's a little hard to go back, so you can do things. We've done a project called Readapt, where we've gone back to old studies that were done fixed, and we figured out what would have happened if you had done an adaptive design. All of those studies have a kind of a built-in bias to them, because it's very hard to get nice apples-to-apples comparisons. So for example, if you did an adaptive design, you've changed the allocation as you go, you can't reconstruct that as a fixed trial anymore. You can't say what would have happened if I had done a fixed trial, because you didn't allocate the patients that way. So what would have happened is more of a random thought experiment than you have that data. So.
One other problem with making a comparison afterwards is that you can, if you know exactly, if there were multiple arms, and you said, well, if I had done a fixed trial, and I had picked this arm that ended up working in the adaptive design, there's always a fixed design that you can come up with later that can outperform the adaptive design if you design it in such a way that you know something. And the idea here is it allows the trial to learn about the uncertainty. After the trial, you say, oh, well, why did he do this crazy adaptive thing and look at these 3 doses? If he had just made up his mind in phase 2 and just picked 2 doses and compared them, he could have done, he could have gotten the same answer. So why did he go through all this fuss? The real thing is that before you start the trial, you don't know. That's why you're doing the trial and building in the adaptations.
So we get this asked a lot by clients. Should I take 1 dose should I skip phase 2 and take 1 dose into phase 3, or should I do the phase 2? And the usual answer to that is, as long as you're never wrong, skip the phase 2. But if you have uncertainty, that's what the phase 2 is intended to get rid of, and to mitigate that risk. So I'm just going to I'm going to skip a whole bunch of stuff.
Yeah, sounds good.
I have to show this. So adaptive trials, this is a standard. So Roger was talking about this slide. It's in every talk. So essentially the goal is talks about pre-specifying. Think of the adaptive trial as a roadmap. The data is meant to guide you on how you're going to go through this. You've got to pre-specify this all in advance. You have to do a lot of thought ahead of time. If I saw this data, what would I do? If I saw that data, what would I do? If I saw this other data, what would I do? A lot of that has to happen, as opposed to just reacting to one data set at the end. So you need this kind of guide. And in the interest of time, because I don't want to go too far over, Roger, can you say a couple minutes on DSMBs, IRBs for adaptive trials?
The basic idea is everything we said about DSMB still applies. The 2 differences are that the DSMB needs to understand the adaptive design so that they can tell if it's being done properly because it's more complicated. And the DSMB needs to understand that they are not redoing the plan for the adaptive design. The pre-specification is just as important. So you want to avoid the rogue DSMB that says, oh, we're allowed to change the randomization proportions, but I don't like that algorithm. Let's do a different algorithm. Or it says to drop that dose. I'd like to keep that dose because those decisions destroy the operating characteristics, and now you have a non-pre-specified design.
The IRB thing I'll briefly cover in that the irony is that if you drop 2 arms through your pre-specified design, the IRB has already approved the entire protocol and all those things that could happen. They may want to know about it, but it's not a protocol amendment. If you change your office phone number on the consent form, you have to submit a protocol amendment to the IRB. So that would be just what I would say about IRBs and adaptive designs.
Yes?
So the FDA, requires a 2 pivotal trial before drug is approved. Is any case, one study using fixed design, the other with adaptive design?
Certainly could, yes.
Yeah. Do you actually have a data to 2 of them that should agree each other, or there's some data that don't agree?
Excuse me?
So if that's the case, do you actually have the data showing 2 designs that agree each other? The conclusion from a different design should agree each other.
I'm going to say I'm sure we do. I don't know if I can come up with an example where one pivotal trial was adaptive and one was fixed.
So, like all good lawyers, we'll answer the question you didn't ask, politicians. There are examples, so there's an entire FDA guidance on when an adaptive design can be considered adequate and well-controlled, which is the regulatory term for when a phase 3 trial is good enough to be one of the 2 trials. Because the actual law says that, with some exceptions, that an approval should be based on 2 independent, adaptive, and well-controlled trials. There is published work that done by our colleagues in which a first adaptive, adequate, and well-controlled trial is running, and then when the data look good enough, it actually automatically launches a second trial. So it chooses the timing of the launching of the second trial adaptively. What I can't remember is whether that trial that got launched was adaptive.
All right. So it's a great